Okay, here we go. So I'm going to start just by introducing the research team. So there's myself. I'm going to put up this slide afterwards at the end of the presentation as well if you want to have our email addresses. But I'm David Beauchesne. I'm a postdoc at Université Laval and also now a postdoc at the University of Toronto. I've been working, I wrapped up my PhD at the end of 2020, and there's a link of this project, a big link of what I'm presenting today with my thesis. And I'm working in ecosystem-based management in general and cumulative effects assessment in particular. And in this project, I was working with Cindy Grant, who is a research professional at Laval University, working with Philippe Archambault, who is the supervisor for this project, and he's a prof at Université Laval at the Department of Biology. All right, so I'm going to start with a little bit of context. So with increasing populations and the increasing demands for natural resources, there's a wide variety of environmental pressures that are exerted on marine ecosystems, be they human activities like shipping, fishing, aquaculture, even coastal development, or climate change related, such as temperature warming and sea level rise. And so individually, each of these environmental pressures can have profound environmental effects on ecosystems, such as commercial fisheries that have induced the global collapse of commercial species, that impose a transition from large species to smaller species and crustaceans, and also deteriorated a bunch of different ecosystem services. Another example could be ocean acidification, which can affect the physiology and the mortality of mollusks and corals, and also alter the flux of carbon and energy in ecosystems. But these environmental pressures rarely occur individually in isolation, and there's a lot of co-occurrence of different drivers in ecosystems. And whenever there's co-occurrence of environmental pressures, there's also the chance for non-additive effects. And so, for example, here we see coral reefs that could be affected by different types of pressures. And it's been shown that, for example, if you have temperature increases with pollution, then the rates of bleaching can be greater than if these two pressures are affected and are present in the ecosystem individually. And so what this leads us is to try to prioritize a bit more of an ecosystem perspective to management that would prioritize research based on our best understanding of ecological interactions and the processes that are necessary to sustain ecosystem composition, structure, and function. And also more of an ecosystem perspective in terms of cumulative effects. And here, for those that are familiar with the literature on cumulative effects, I'm not talking about that what's done in cumulative effect and environmental impact assessment, which is much more interested in projects, in particular projects and activities, but rather something that's more akin to regional cumulative effects assessment, which focuses much more on understanding the interactions between human activities and the natural world. And so in 2008, there's paper by Halpern and colleagues that was published that offered a very nice approach to that was more an ecosystem perspective to cumulative effects assessment. So they did a first assessment of the footprint of cumulative human activities on the global oceans, first in 2008, and then an updated version in 2013. And so this is what you see on the first row up there. And so what they essentially found, well, you see like whatever is in blue, like the colder colors identify areas of low cumulative effects. And when you go to the red, there are areas of higher cumulative effects. And what they found in a nutshell, essentially, is that there are very few areas of the globe that are free of cumulative effects. And below, what they show is the rates of change through time, and they showed that through time cumulative effects have tended to increase during that time span. Now, as interesting as this method is, in terms, when you're interested in species, it does not consider one of the important aspects of species, which is species interaction structuring the ecological communities. So species don't exist in a vacuum; they're actually embedded in complex systems. And this gives rise to indirect effects propagating through species interactions. And so there is a classical example here that I show between sea otters, sea urchins, and kelp forests, where if you decrease the abundance of sea otters in the system, it's going to lead indirectly to a decrease in kelp forests because sea urchins are going to take over and browse on kelp forests. And so these types of indirect trophically mediated indirect effects can be quite significant, and they can be at least as important as the direct effects. And they can lead to the propagation of the effects of environmental pressures that will be a function of the structure of the network and the position of a species in the ecological network, so that, for example, the predator is not going to be affected the same way as a resource. And so this was the topic of my thesis that I, like I said before, that I wrapped up at the end of 2020. And so what I did is we expanded on the Halpern method to develop what we call a network scale cumulative effects assessment that explicitly considers this network of species interactions that allows us to consider both the direct effects of stressors on species, but also the indirect effects arising from species interactions. And so we did this for around 180 species and 17 environmental drivers in the St. Lawrence system. So I'm not going to spend too much time on this particular result and the results from my thesis, because essentially the goal of this project was to emulate what I did during my thesis in the St. Lawrence, but to do it in on the Scotian shelf bioregion. So the general objective of this project was to assess the cumulative effects of global changes on ecological communities of the Scotian shelf region. And we had 4 specific objectives for this project. The first was to characterize the intensity and the spatial distribution of environmental drivers, so for example, shipping and fisheries. Then characterize the spatial structure of ecological communities. This means the distribution of species as well as the interactions that structures the ecological communities on the Scotian shelf. Then we had to assess the vulnerability of species to environmental drivers, and finally put all of that together to assess the cumulative effects of environmental drivers on species. So, like I said, the spatial extent of this project was the Scotian Shelf bioregion. So it included the Bay of Fundy, and to the east, which is south of the Laurentian Channel. We divided the whole study area into a regular grid of approximately 1 kilometer square, and we divided the temporal scope of the assessment in 2 distinct periods of 2010, 2015, and 2016, 2021. Now, the results of those 2 periods are qualitatively similar. So today, what I'm going to be presenting is only for the second, so that for 2016, 2021, but in the actual technical report that's available for the project, all of the results are also available. Okay, so now I'm going to present the assessment method. And I'm starting by presenting the Halpern method, which I'm going to be referring to as the species scale assessment. So imagine that we have a system composed of 3 different environmental drivers, which would be temperature increase, shipping, and fisheries. And system composed of 6 different species, so humpback whale, beluga, cod, capelin, krill, and copepods. So in terms of assessing cumulative effects with the Halpern method, what you would do is first you would focus on all of the different individual combinations of environmental drivers and species. So for example, here, the effect of temperature increase on copepods. And what you would do is first what you would need is to first have an assessment of the spatial distribution and intensity of the driver that you would multiply with the distribution of your species, that you would then multiply by the sensitivity of that particular species to that stressor, which we term the species-specific sensitivity. And then the result that you would get is a map of predicted effect, individual effect of that particular driver on that particular species. And then the game is to essentially do this for all possible combinations. For example, here, as columns, we have different stressors, drivers, and as rows, we have the different species that we would want to consider, and you can evaluate the 9 different possible combination of driver species effects to have one map per species per combination, and then you would sum all of these together to obtain the evaluation of cumulative effect, which is the Halpern method. So what's nice with this method is first you get an aggregated evaluation of cumulative effect, but you can also look at all of the individual effect map if you're more interested in individual components of the assessment, or even like subsets of different drivers or species. So it's quite powerful, and the ability that you have to afterwards explore the results that come from this method. And so this is the Halpern method, or the species scale assessment. Now, what we did is, rather than consider all species on a level plane and not interacting, we expanded this method to be able to consider the indirect effects arising from ecological interactions. And so if we were to try to characterize a complex network, it would be quite difficult. And so what we did rather is focus on motifs, which are submodules of a certain number of species that you can use to deconstruct and simplify a complex network, and that represents structural properties of your full network. You can study those submodules and then put them back together to get an idea at the full scale of the network. And so we focused on 3 species motifs and on 4 in particular, because if you were to take an empirical food web and deconstruct it in all the 3 species motifs available, you would find that they would be composed between 90 and 95% of the 4 different types of 4 single types of interactions. And those 4 are tritrophic food chains, omnivory interactions, exploitative competition interactions, and apparent competition interactions. And so these have been the focus of the approach that we developed to assess cumulative effects. So how do we do this? So going back to this graph here, we start off the same way as the Halpern method, where we look at, for example, here, if the focal species is cod, we would start by looking at the direct effects on cod. So here, the direct effect of fisheries on cod. But we would also consider the direct effects of drivers on the other species it's interacting with. So here, if we look at the omnivory interaction between beluga, capelin, and cod, we would also take into consideration the effect of fisheries on capelin and the effect of shipping on beluga. And then we weigh this assessment, this cumulative effect score on this interaction by what we call the trophic sensitivity of the focal species. So essentially here, beluga, capelin, and cod are not vulnerable to the effects, the indirect effects propagating through interactions in the same way. So typically resources would be less sensitive to indirect effects than top predators would be. And so this is what we term the trophic sensitivity of our focal species. And so this gives us an assessment of cumulative effects for this particular triad of species. And we would do this for all of the different three species interactions in which cod are involved. So there's 4 here, and take the average, which gives us a score of cumulative effects for cod that takes into consideration both the direct effects of fishing on it, but also the effects of the other drivers on the species it's interacting with and its trophic position. And so we can do this for all the species in the network, and we aggregate all of these together to get a community aggregated assessment of cumulative effects. Okay, so now that I've presented the method, I'm going to discuss a little bit about the data modules that we used for this assessment. So the actual assessment revolves on 5 different data modules that we need to gather to be able to do the assessment. So I'm going to present them and go through them rather quickly because each data modules could be an individual report or an individual paper. And so what I'm presenting today might open up more questions than it answers. But if you have any question, we can talk about it at length during the Q&A. So the first one that we need is an assessment of the distribution and intensity of the drivers in the system. So we did this for 16 different environmental drivers. So we have 4 that are climate related, 6 that are coastal, 5 different types of fisheries, and 1 that is related to shipping, essentially. And if anyone went to the link that I posted earlier and you're following along, you'll see that whenever there's results that I'm showing, you can click on it to see the figures on their own and be able to zoom. And whenever you see the book here, you can click on it and it's going to take you to the part of the report that relates to what I'm currently talking about at the moment. Okay, so the second data module that we need is the distribution of species in the system. So we ended up characterizing the distribution of 172 marine species, so mainly fish and invertebrates, 8 marine mammal species, and 25 species of seabirds. The third one that we need is the species-specific sensitivity. So all of these 3 essentially are what Halper needs to perform the assessment. So how we went about evaluating species-specific sensitivity is a trait-based approach. So for each species considered, we evaluated a series of traits, which is body composition, environment, feeding mode, mobility, phylum, and size, which we related to the different environmental drivers that we considered for the assessment. And so for each driver trait combination, we gave a value between 0 and 1, 0 being a species that is rendered insensitive because it has a certain traits, and 1 being the maximum sensitivity. And then for a particular species, we would do the product of the relative sensitivity of all the traits considered, which gave us a relative sensitivity for each species that varies between 0 and 1. So what this means, for example, for shipping and minke whale, the traits that we considered for shipping were environment and size. And for minke whale, the assessment essentially would be that it's a pelagic species, which gives it maximum vulnerability, and it's over 3 meters, which also gives it maximum vulnerability. So its overall sensitivity to shipping would be the product of 1 and 1, which would equal 1. And so we did this for all the 200 species considered and all of the different, the 15 environmental drivers considered to get an assessment of species-specific sensitivity for our assessment. Okay, now we get into what's needed for the network scale assessment. So the first data module that we add to what Halpern did was characterization of the meta web. So this is the characterization of who eats whom in our network, so the food web. We did this using an unsupervised machine learning algorithm that basically goes through a series of logical steps to predict the binary interactions between species under the assumption that similar species would consume and be consumed by similar species. The similarity criteria that was used was taxonomy and diet. And we used a interaction catalog of empirical interactions that came from a variety of different sources that yielded essentially over 180,000 pairwise interactions in marine environments for close to 15,000 marine taxa. And the last bit of work, the last bit of data, the last data module that we needed was the trophic sensitivity that I referred to earlier. So basically, the sensitivity of a species to the indirect effects based on its position in a food web. And so I bring you back to the three species motifs here. And essentially what we did is we simulated stress on these four different types of interactions using Lotka-Volterra models. And what we did is to simulate stressors that we applied a 1% variation of equilibrium parameters on these equations. And we simulated all different possible combinations for each motifs. So that varied between 127 to 511 different pathways of effect through which these interactions could be disturbed. And we measured the initial and post-stressor abundances that we used as a proxy of trophic sensitivity. And this is what we're using as our trophic sensitivity evaluation for the species in the food web. Okay, the results. So I start here by talking about species richness and then only the cumulative footprint of drivers. So essentially, this just aids into the interpretation because the actual assessment, the spatial assessment, is governed by the co-occurrence, obviously, of drivers and species together. So looking at them separately kind of helps understand the cumulative effects results a little bit more. And for each of the following maps, the color scheme is always going to be the same. So dark blue means no to low values. So here, number of species. And when you get to yellow, then these are the maximum values. So in terms of species richness, what we found is that there's medium to high species richness on the continental shelf. And then eastern and western Nova Scotia, and especially the western Bay of Fundy. And then there is a bit of line at the transition between the shelf and the slope. And then we see lower species richness when you go to coastal areas really close to the coast and continental slope. Now, I just want to put a caveat to that. The data that we use essentially was much more abundant for the continental shelf itself than the slope and the coastal areas. So this shouldn't be interpreted as these areas are devoid of species, but rather a product of the data that we use for the assessment. Now we have to look at cumulative drivers. So essentially what this is the sum of the intensity of all of the environmental drivers that we considered for the assessment. And so we see that there's medium-high cumulative drivers on the continental shelf, the eastern Bay of Fundy, and the coastal areas, especially close to coastal towns, and low cumulative drivers on the continental slope. Once again, same species, we have much less data for the continental slope. In fact, there's only 3 drivers that extended all the way to the continental slope. So we need to be careful when we're interpreting the results for the continental slope. Okay, this is the actual result for the cumulative effects assessment. So what we see is that there is high risk of cumulative or high predicted cumulative effects, especially in the Bay of Fundy, then southwest Nova Scotia. So this whole region here, then here northeast of Sable Island, which is located just around here. And I hope you guys can see my my mouse here. Otherwise, you're not really understanding what I'm doing there. And then the south of Cape Breton. And then you have medium cumulative effects at the center of the shelf and the northeastern corner, and very few to no effects on the continental slope. But once again, I want to remind you that we have very few species there because of the data that we use, and also this very few drivers that were characterized in that location. So I present this figure here as a way to provide an overview of all the data that's accessible through this assessment. So what you're seeing here is a meta network that presents essentially all of the different links, which are pathways of effect that were predicted through our assessment. So if you look at the upper right quadrant here, like in the 4 different colors here, you have all of the different stressors that were considered for the assessment. So marine traffic, different types of fisheries, coastal drivers, and then climate drivers. And here you have invertebrates classified in bigger groups like arthropoda, echinoderms and stuff, and mollusks. And here are the vertebrates, where you have fish here, marine mammals, and marine birds. And so the size of the points reflect the overall, either the overall effects of drivers or the overall effects on species. And each line represents a predicted effect between driver and species. And so we don't want to interpret this figure in a really granular way, but it does provide a nice overview of all of the different links that are predicted and all of the different links that could be explored individually or in group through this assessment. There's still a few things that can be gleaned from this figure. So for example, we clearly see that a lot of the effects come from shipping, one fisheries type, and one type of climate driver. And that in general, you have a bigger density of effects on invertebrates than on vertebrates. So now I just want to show the links that arise from direct effects. So discounting species interactions, what would we see if we only focus on direct effects? And so we see that there is much less links. So there's about close to 900 different pathways of effect that are found, and we see that the density on the invertebrates doesn't change all that much, but we see, for example, here the effect of shipping is much lower, and it affects much less species. And then if, for example, we look at birds here, we see that if you only look at direct effects, you see that there are very few links between the drivers that we considered and marine birds. And so if now instead we look at the indirect pathways of effects, we see that, for example, here shipping is much more prevalent on all different species than it was before, and birds as well now are much more affected indirectly. So this was to see this at a glance, but we'll look at it in a little bit of a different light now. So what we're going to see here is the contribution of drivers to direct and indirect effects. So this is going to be a stacked bar plot. So what's up here is going to be the direct effects on invertebrates and vertebrates, and the different colors relate to the different types of drivers. And also here, the different groups are found. So if we look only at direct effects, what we see essentially is that the invertebrates are affected directly from all drivers. And this makes sense. The invertebrates are sessile species. They're, in general, more sensitive to their physical-chemical environments, which makes them more vulnerable to the direct effects of drivers in general. And then if you look at vertebrates, then the outlook is quite different. There's much lower effects, or at least the diversity of drivers affecting vertebrates is much lower, especially for birds and marine mammals. So birds is a little bit of fishing and mostly shipping for vertebrates. And so This makes sense in the sense that usually vertebrates or mobile species that have the ability to minimize exposure to stress. But if we look at indirect effects, if we overlay it here, we see something quite different. So in terms of invertebrates, it doesn't change too much. They're still affected by all different driver types, usually at a similar relative intensity. But there isn't such a big change from direct effects. And so if we look now instead to vertebrates, we see something quite different, especially for birds and marine mammals, where we see that now they're affected by all types of drivers and indirectly, and sometimes at greater relative intensity to that of direct effects. This can be related essentially to the fact that these species are usually occupy higher trophic levels, and this makes them more sensitive to indirect effects. And I'm going to show this graph here to show why it could matter, especially in the context of management. So if I open only this figure here, so what you see here is the continuous distribution of minke whale, and this is the binary distribution that we used for the assessment. And in the report, there's an atlas that has one figure like this for the 200 species that were considered in the assessment. If we move to this figure here, so let's focus only on the first period here. So what you see is, sorry, I keep zooming and unzooming. I'm trying to get this right. So this is at the middle here, the assessment you would get if you considered only direct effects. And essentially what you see is the shipping lanes on the Scotian shelf. But if you consider species interactions, then the effects to minke whale are quite different and located in different areas than what's predicted if you were to look only at direct effects. So this could have significant impact when you're looking at the management of a species if you're essentially looking only at what's obvious, which are the direct effects, but ignoring the potentially significant effects that arise from indirect effects on the resource of that species, essentially. Okay, I'm going to go over some perspectives now before I wrap up the presentation and focus on two particular perspectives. The first one is about the data modules. So the whole assessment is, like I said, predicated on 5 different data modules. And what's interesting is that those are very modular, no pun intended, and easy to update. So each of those could be independently refined or updated or completely changed if we had better data and then the analysis rerun. So for example, if an expert on cod was to look at the distribution of cod that we're using for the assessment and say, this map doesn't make sense, then we could essentially change the map for cod itself, rerun the whole assessment, and update the assessment quite efficiently. And this can be done also quite efficiently for reasons that I'm going to get into in the second perspective. But this gives you, essentially, in my mind, gives the opportunity for additive management and periodic assessments that essentially can be updated and made better through time because the data that we have also is getting better through time. Another perspective that I don't want to forget is in the same area, there's a team led by Noreen Kelly who did an independent assessment of cumulative effects, but rather than focus on species, they're focusing on habitats. And something that would be quite interesting would be to compare those 2 assessments together and maybe even combine them together to have a joint assessment of species and habitats together. Now, we've already exchanged notes and we've seen that both of our assessments are quite consistent. So the areas of the Scotian shelf that are deemed as more affected by cumulative effects are roughly similar from between our assessment and their assessment, even if we're not considering the same valued components, so them habitats and me species. But it would be interesting to formalize that and go a little bit further. And sorry, sip of water. And the last perspective and what I would like to wrap up my presentation with is about data science and reproducibility. And this is more than a perspective for me, because I've been putting in time for the past couple of years to really make sure that everything I do is as reproducible as possible. And I'm going to spend some time talking about this. I've worked either through my postdoc or my company with DFO, with Environment Canada, with Transport Canada, to try to make processes more reproducible. And in my mind, this is the way that we're going to be able to do efficient science that is going to be more centered on actual decision making rather than data gathering, which usually takes like 90% of the time whenever it comes to ecosystem scale assessments like the one I'm presenting today, but also analyses like marine spatial planning, spatial strategic conservation planning, like Marksan analyses and stuff like that. A lot of time it's dedicated to gathering data, and there are ways to make this much more efficient. This is what I'm going to be talking about now for a little bit. So, like I said, there's a lot of data that's required, that's a vast knowledge that is required for these types of assessment, and they're typically distributed between many different experts. And the result of this is that there's a lot of time dedicated to researching relevant information for an assessment, developing collaborations, and formatting and structuring the data that are needed for the assessment. And so we need to strive to facilitate the reuse and secondary uses of the data. And so it's partly why a lot of the data that I'm using for this assessment are openly available data, for example, in open government that are easily accessible. So right now there's an important movement towards openly sharing knowledge, and open government is a good example of archiving and online access. So it offers a number of different advantages. It allows you to make processes that are going to be more transparent, efficient. It can also optimize investments, promote knowledge, and promote collaborations. And so this is the way to go, forward. I would also make the case that usually when we start on a project, so for example here, if I do cumulative effects assessment, there's going to be a back and forth between environmental management and cumulative effects assessment, but usually the process of gathering knowledge or gathering data is going to be part of the assessment itself. And I would argue that the knowledge gathering should be external to the actual cumulative effects assessment or ecosystem-based management process, and that we should dissociate this to provide better efficiency, a common structure, and accessibility, which would foster collaboration, mobilization, and like consultation, and it essentially would allow us to focus on the management and assessment rather than gathering knowledge. And so here is an example that was also part of this project, which we call eDrivers. eDrivers was initiated for the St. Lawrence, and it was essentially an open knowledge platform that seeks to share knowledge on environmental drivers in the St. Lawrence. And the objective was to create a community of experts dedicated to structuring, sharing, and standardizing knowledge on environmental drivers in the St. Lawrence to begin with. And this was done as part of the Canadian Healthy Oceans Network. But now we extended this. So if you go to, this is the actual application. Now there's an additional tab that can be found where you have the Scotian shelf. And each individual drivers that we considered for the Scotian shelf can now be explored interactively. So either in isolation or combined with other stressors. And essentially, if you select them all, it's going to give you the same figure that I showed earlier about cumulative drivers. And so these can be explored um, interactively, and they're openly accessible now through eDrivers. Okay, now I'm going to hit you with a bit of a metaphor, and you can let me know in the Q&A whether it works or not. But essentially what I've been describing now is data archiving. And right now we're in the era of data supermarkets. There's a lot of different platforms that are out there that offer lots of different data. So I view this as walking into a supermarket where there's a lot of products available. Some are raw products that you can have access to, but there's also some refined products, and you're not really certain of the quality. Some have labels, some don't. But this is what we have, and so this is super 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 important to have. But if I was to ask you to cook me a 6-course meal, then it would be much more difficult for you to do. And so my question would be, where is Ricardo? Where are the recipes that we need to be able to efficiently use the different ingredients that are available in our data supermarkets to be able to produce the different data sets that we need, and even the different analyses that we need for more ecosystem scale assessments. And so what I'm talking about here, when I'm talking about recipes, is essentially reproducible analytical pipelines. And so reproducible workflows are super interesting and important for a variety of reasons. But most of all, they are accomplished by using programmatic tools. So for example, what I'm using personally is R and GitHub. And essentially all of the different steps of a project, be it accessing raw data, formatting data, the analyses, figures and tables, the report, and even the presentation that I'm giving right now, can all be programmatically constructed. And if everything is done programmatically, then you can have a full pipeline that's going to allow you to reproduce a complete project rather than only individual parts. And so there's a number of advantages in working like that. It allows you to have a process going to be structured, reproducible, and transparent. You can reuse workflows across projects. So for example, if the characterization that I did for shipping is of interest for another project, they could essentially just reuse the reproducible pipeline that I prepared for shipping and use it for their project. They can also be used to update projects. So if shipping changes, I can just change the pipeline at the beginning and rerun the whole thing and be able to update the analysis. And so the main advantage of this is an increased efficiency, and we're talking about months and weeks to minutes. And there's a bunch of different examples internal to DFO that I think a few people in the audience could attest to. But I'm going to give one particular example from this project. So in early February, I did something stupid. I deleted the whole project that took me 2.5 years to prepare. I don't know how. I know how, but I don't know why I did that. But essentially, I pressed enter and I lost everything. If I had not worked with data science and reproducibility, I would have been like the project would have been screwed. But because I do have a reproducible process, within half a day, I was back to the exact state I was in before I pressed enter by mistake. So this is a really, really strong incentive in my mind to start thinking about data science and reproducibility. And so for one of the product that's available through this project is a research compendium that's called NCEA DFO. So if I go here, essentially what you have is all of the code that was developed to produce the assessment that I presented today. And so this is all available on GitHub. And there's like, if you go on the website, you are going to have a description of the content and how to use it. Also, if you go to the link that I showed earlier, essentially you have the 4 different products that have been created so far for this project. So the webinar is what I've been presenting today. eDrivers, we just had a quick demo earlier, and the compendium is what we watched, what I just showed you. But you can also have access to the current state of the technical report that comes with the paper, and there's much more information in the report than what I showed today. And so you can access this through this link. So this is the first, let's say, reproducible product that was created, and the main one. And then the other one, throughout this project, I started thinking about ways of formalizing and sharing analytical pipelines that I've been creating for this project. And so I started a project called PipeDat, which essentially is an R package that allows a user to execute data pipelines that grant data access and formatting, and greatly facilitates the creation of integrated data sets. So essentially what you need is an ID of a specific pipeline, and you select all of the different pipelines that you need, you execute everything, and it's going to fetch the data, format it, and if relevant, integrated into a study grid for further assessments. So this is a work in progress. I'm hoping that we can continue working on this. It's not formalized yet, but it's something that I started thinking about, and that the NCEA DFO research compendium relies on to get the data. But there would be much more that we could do with this package. So, for example, here, the combination of the research compendium and the PipeDat package. What you see here is a table of globally all of the data sets that were used for this, a condensed version of all the data sets that were used for this assessment. And we see that everything that was used for the Scotian shelf, and most of the data would also be available directly for the St. Lawrence. Now, the St. Lawrence assessment is already done, but I'm just showing here what it could be used for. So it could be, most of the data could be used for the St. Lawrence and also for Newfoundland and Labrador. So what this means is that the combination of the research compendium and PipeDat already puts us on the doorstep of being able to do an assessment of cumulative effects for Eastern Canada. That would be much more efficient than if we were to start from the beginning. We can start already with what's already been done for the Scotian shelf. So to wrap up the presentation, I'd just like to thank all of the collaborators and organizations that exchanged and shared with our research team, be it for their expertise with data or to share data sets. And specifically also the Department of Fisheries and Oceans for giving us this contribution agreement, and that allowed us to work on this project for the past 3 years. Particularly to Tana Worcester, Caroline Lantin, and Catalina Gomez, your presence and your support and guidance was really, really crucial for this project. And finally, Jacob, Andréane, and Vincent, who helped throughout this project. Thank you. If you have questions or comments, you can reach out now in the Q&A, or you can take our emails and reach out to us. It's going to be a pleasure for us to have more discussions with you guys. And now I would open up the floor for questions.